So a big part of this course is going to be getting some practice at not only identifying plants in the field, but also making permanent records of what's out there. So students in this class can collect plants, and as a process, they collect them, dry them, and we keep them in basically a plant museum called the herbarium, where they can be used for years and years after this as a record of what's out there. So this is not only just it might seem kind of like a hobby to go out and collect plants, but it's actually a very important part to many applications of botany, including ecology and taxonomy. So nice introductory image here with a plant in the foreground and some botanists going around maybe locating more individuals of these plants. So when I say specimen, a specimen is an example, like a single individual, an instance of a species. So specimens end up in collections. So like I said, our collection is the herbarium, where we have a collection of pressed and dried plants. So these are some images of pressed and dried plants. These are from the University of Connecticut Herbarium, where they're actually available as digital images. So this is something that we can access from anywhere in the world. You could access these images, and they're actually at really high resolution. The little zoom ins on the labels don't do these justice. You could zoom in quite far onto these specimens and get a lot of detail from them. So they're useful for so many reasons, and I'm hoping to show you some of that as we go through this. So it's not only an instance of where and when a particular plant was growing, but all sorts of features about that plant. So sizes of organs and reproductive status and so on. In addition, so collecting a specimen is a good way to really get a scientific proof that you have seen the specimen that you said that you saw. So if you go out and you collect data about what plants are growing in a habitat, if you bring back examples of the species you found, then you could say, yes, I found this species, and here it is. Here is one of the individuals that I brought back as a permanent record, as a so-called voucher specimen, showing evidence of that species being there. Another bunch of information. So these are not just the plant material themselves, but also a collection of information on the label about what was known about the site at the time. So you can find out features like we have fruit color here, arching habit of the growth form, location, some over here, features of this plant, an annual plant. Sometimes this mentions a river. Sometimes there'll be information on here about the habitat, so light levels and soil type and so on. So lots of extra information besides just what's contained in the plant itself. So some of the things we talked about already owe themselves to plant specimens. So when a person gets really familiar with plants in Wisconsin, if you want to write a book where you tell people how to identify plants in Wisconsin, you probably wouldn't have a collection of live plants in front of you. You need to use some sort of resource that allows you to collect a bunch of information in a short amount of time, right, with minimal effort. So you could go to a herbarium where they have thousands of individuals of any one species. It might be an over-exaggeration, but tens or hundreds of individuals of the same species. And you could do something like we've done here to take measurements. So this study was one that I was an author on. We took plant specimens, hundreds of plant specimens of these two species in the genus Nymphoides, and measured parts of those specimens. So we had leaf sizes and petiole lengths and all kinds of things on there that could be used in a kind of a big data approach. So lots of measurements from lots of specimens across lots of places and lots of times and different years, different times of year. All this information to get kind of a comprehensive view of the species itself in order to answer some of these questions like how big can they get? How small can they get? Are they always different in this measurement, or do they overlap sometimes? So this is part of the projects that I did for my PhD. This was a project that did not have me driving around through the east of North America to find these species. I could just take loans of herbarium specimens and take measurements on those. So a lot of value that still can be grabbed from these specimens. And I have some research where I still do this kind of thing today, where you can learn a lot about species without going to find them out in nature. Some of the things we use, so our dichotomous keys, are informed by information that largely would have been obtained from plant specimens. So just like I showed you on the previous slide, knowing the ranges of variation, the maximum lengths, the minimum lengths of organs, presence or absence of something like pubescence, hairs on the epidermis. If you get a comprehensive set of specimens, or you've seen the species across its entire range, then you can really draw a good solid conclusion about what features are present in that species and what features are never found in that species. Also could be a useful tool for identifying new species if that ever came up. So if you were studying plants specimens and found a feature that was not consistent, or maybe features that were consistently different within what you thought was the same species, you might actually have a new species. So lots of opportunities for studying specimens. Here it's handy because this is something we can study in the off season outside of the field season. Another feature, so all these places where plant specimens were collected all together can be used to build one of these range maps. So if you ever see a map of where a species can be found, a lot of times these maps are drawn, at least in the past, would have been drawn exclusively from specimen data. So you take your collections and say, we got this species of beech, and I know every place that it's been collected, all these dots, different habitats, different states, and you zoom out to the east of North America, and you can plot them on a map and say, these are areas where this species has been found. This is where it can reliably be found still. And then you draw a map. So on the left here, this is data from the GBIF resource, the Global Biodiversity Information Facility. And the maps don't align exactly, but they have quite a few features in common. So you see the data points here in the east of Wisconsin up through Upper Peninsula of Michigan. Is same distribution as here. So these are on the left dots of specimens, and on the right just an artist's scientist's rendition of the range limits of these species. So it takes in this case, hundreds of specimens to make up the underlying data for a map like this. So I think when I was younger, I didn't really think about where we got our distribution maps from. But people don't just dream these up. They have to build them using data, and this is the kind of data you get for those. So I mentioned voucher specimens. Voucher, so if you think of somebody vouching for you, if you have an alibi and somebody vouches for you, you say, I saw this person and they were at the restaurant last night. Same thing with a plant. If you say, I saw this species, scientifically, we should be able to back up our claims with evidence, and this is evidence for finding something in a place. So if you go to a habitat and you write down that these species are present, if you make a specimen of your species, then you have evidence that that in fact is growing there. So if you were to do an ecological study, you might actually end up collecting specimens to demonstrate, without just saying that you have the names, but actually demonstrating the names for the species that you've got. You'd be collecting your voucher specimens. And that's one of the original uses for specimens. This kind of technique goes back hundreds of years. And we have, still today, we have specimens that are 300 years old or more in some parts of the world. So it's a good, reliable way to preserve a specimen into the foreseeable future. So good evidence for what you've seen. And I'd say the second point, vouchers being most important, areas where you might have an exceptional claim. So let's say you are the first person to find a plant species in a country or a county. Those are a little more exceptional. Somebody says, well, we didn't think that plant species was there. Are you sure you found it? And just telling them is not maybe good enough, as certainly not as good as showing them the physical specimen for what you found there. A little bit more, say, everyday kind of uses for voucher specimens. Just sometimes useful to do an inventory, sometimes periodic collections of the same species can be useful. So we have, for our herbarium, we have students collecting specimens, and we just sort of add in the specimens as we get them. If they're good quality, they end up in our permanent collection, and we end up over time getting this more comprehensive perspective of what grows in a particular place. So here's an example of an ecological study that just wanted to point this out. This really highlights the specimens that are underlying this kind of study. So this is a alpine plant community in New Hampshire, so growing on mountain habitats, and a variety of species collected there. And this might be a habitat that not a lot of people have been to. It might be hard to access. And so it might be an area where you might just casually not have people who know what's out there. So you can show them by depositing these specimens into the herbarium. So all of these abbreviations here, this CON is the abbreviation, the official abbreviation for the University of Connecticut Herbarium. Ours at UW-Whitewater is UWW. And then there's a unique number that follows that. So this is I think like a barcode number. So 139484 gives you an idea also of how many specimens are there. So hundreds of thousands of specimens contained at Connecticut Herbarium. Our herbarium is up at about 7,000 right now by comparison. So we're not a large herbarium, but we got some important plants there. So here's some images of what the actual herbarium looks like. So pressing and drying plants, this is a technique, like I said, that goes back a long time. Most plants are pretty amenable to this kind of treatment. So you take the flat part of a plant, typically the leaves and reproductive parts are pretty useful for identifying plants. And if you press them and dry them, they end up being pretty flat for the most part. So flat plant specimen is something that you could stack in pretty high density in these cabinets. So this is a herbarium cabinet. The specimens are placed into folders and organized by species, by genus, by family, basically so that you can find what's there, right? So if you need to find your specimen, knowing where to go to find that is useful. And for example, here's a couple of plant families, these big cards here showing different plant families that would be referenced in this collection. The cabinets are pretty standard. They seal up tightly. This is to keep, they want to regulate moisture levels, so plants should stay dry. And also pest control. So there's a variety of pests that will chew up plant material, and they have to keep them out and sometimes treat for them. You can use chemicals to treat them or even just freeze them off. That's the technique we use. All right, so pressed and dried, archival paper, archival labels, archival glue that keeps these from degrading over time. Sometimes old specimens will have acidic glue or something that ends up sort of dissolving the paper over time. Paper sizes are generally pretty standard. We have just about all of our specimens are the same size paper. And then the cabinets, as I mentioned. And I like these images. A lot of times the specimens just get put away and forgotten, but you know, if you're gonna hold on to something for hundreds of years, just looking at it every 5 years or so is still pretty valuable. So these bigger collections, like at UW-Madison and larger institutions, they get a lot of use. So if you're going to make a plant specimen, it's important to kind of align your collecting strategy with the proper techniques. So make a specimen that is suitable to put into a herbarium, and also, you know, good quality, just one that's going to work out as you dry it. So some images here showing the process kind of going from left to right. So this looks like possibly kind of a local, maybe a little more tropical locale. People out in the field, looks like they're identifying at the same time that they're pressing. The pressing process, pretty much in every place where I've experienced it, involves putting the plant into a sheet of newspaper. So newspaper is pretty handy as a readily available, also disposable, also kind of like fine pores on the paper. Good system for having like a surface to press the plant against, and also a place to hold it temporarily until it gets mounted onto paper So during the process, before getting to the point of pressing the plant, it's important to go through the process of identifying the plant. That's a strong recommendation from me, is to identify your plant before you get it in the plant press, while you've got fresh flower material, leaf material. If you're lucky, you've got additional material, maybe some expendable plants nearby. You can take their flowers apart and press another one where the flowers have not been taken apart. The notebook here indicates that you should be taking notes about the habitat, so light levels, soil type, species abundance, location, water availability, things like that, plant features. And then here, this is a plant being pressed onto newspaper. And on the right here, this is the final product that I've shown you a few times. The final product basically amounts to grabbing this plant after it's been dried and pasting it onto a hard archival piece of paper, and then adding a printed label with accessory information. So the plant press is shown here. This is just a hardwood lattice with cardboard spacers to let some air through. And then after collecting plants, this will be placed into a dryer. So just like a plant cabinet that's got hot air moving through it to dry those out as quickly as possible. So if they're dried quickly, they end up being pretty lifelike, and that can stick around for many years. So we have some very green plants in our collection that are 50 years old or more because they were dried well. Okay, so what do you collect for a plant? That depends on the size of the plant. So it's a little hard to give you an example of every possible way to press a plant. So I've got some indication of maybe the variety of plant material you might run into. And I'll show you this and other slides. These are images courtesy of the New York Botanical Garden, another very big herbarium. Generally, you want to get plants that are in reproductive condition. So plants with just leaves, sometimes you can't identify them if they're vegetative only. And then just generally, it's a little more comprehensive to have a plant that's got flowers or fruits for the flowering plants, or if you've got a conifer, cone or something on there. These are additional features. You got sort of the maximum output for the effort of making one of these specimens if you've got some flowers included. And then trying to make the material you're pressing representative. So obviously it's going to be flat, but as much as you can, try to make that flat appearance look like the plant did in life. So if an organ is erect, stiff, upright, put that in that same orientation. If something is drooping, then make that droop. For example, we've got here some flowers that are maybe a little more dangly on this one. They were pressed in that way. This flower goes up and then turns off to the side, so that's pretty good representative pressing there. Leaves, because this is pressed in 2 dimensions, it's important to try to get the upper surface and the lower surface of leaves. So we've got here under surface and an upper surface. Basically all the representative parts you can get into one of these specimens. So I've got some examples just sort of working through. I have a handout that you can download for making your own plant specimens. But these are just some illustrations of different kinds of plants and the considerations that go along with each kind of plant. So this first one here is woody plants. And woody plants are large, right? So this standard herbarium sheet size of about 16 inches, that is not big enough to press a whole plant on, right? So you're gonna have to get a representative part of the plant. And here's where that feature of plants being modular comes in handy. So no matter what time, what year you get your specimen, no matter what part of the plant you get your specimen, for the most part, you should be getting a basically representative part of the plant that's more or less the same no matter what part of the plant you got it from. So this little branch of this blueberry bush, it's good enough to give you an idea of what the rest of the plant looked like. So you would need, as part of your field equipment, a pair of snips. So trim this down to a size that fits. A little bit difference in the orientation of some of these. So maybe a branch coming off from the side. Maybe that was just the best way to make this fit on here. And then other considerations that we did before. So upper and lower surfaces of leaves, reproductive organs present. And in this case, since you're not pressing the whole plant, you want in your notebook to record something about how big the whole plant was. So anything that you can't see as part of the specimen will be important to include. So is this tree 30 meters tall, 5 meters tall, or anything else about it that's noteworthy? On a woody plant, maybe the characteristics of the bark, something that you can't see, or branching patterns or something like that. Not too different from the previous one would be herbaceous plants that are also too large to fit. So the whole plant, it's understood, would be bigger than the sheet, and you want to get a representative part of the plant. So some sort of reproductive tip from these plants. And I think it's generally understood that if a plant species gets to this kind of size, you don't typically have like roots or underground organs included as part of the specimen, or leaves coming out from the base of the plant or anything like that. So you got kind of your representative tip, reproductive tip of the main stem or a side stem. And same considerations as before. Take some notes about what the whole plant looks like. How big is it? Are there different kinds of leaves outside a frame that we weren't seeing? Is there anything different about the characteristics of the epidermis toward the base of the plant, something like that? All right, moving down in size. These are herbaceous plants that do fit onto a single sheet. So your plant press is sized about the size of a sheet of herbarium paper. So if it fits in the press, it should fit on the paper. A few of these, let's see, the two middle ones can be pressed basically start to finish in kind of a diagonal. You can see some roots coming out of here, some discoloration showing that that was the part of the plant that was at the ground level or underground. So we've got basically the whole plant here. This plant here, we've got a root and some rhizome there. And so those fit without bending anything. The two on the outside have been bent, and that's a standard solution to plants that are basically skinny but tall. So grasses, for example, quite often get this treatment where they're zigzag bent. And the zigzags are deliberate. So bending at a sharp angle tells the viewer that this was deliberately done, and it makes sure that nobody's confused about this plant maybe accidentally or on its own having a zigzag or drooping feature. So you don't want to say make a gentle bend where it kind of swirls around in a circle, but instead make a sharp bend. We've got roots down here. So we've got zig, zag, zig, zag, and then the top of the inflorescence there. That's handy because having the whole plant on here in a zigzag actually contains the information for how tall these plants are. So you don't have to record how tall they are. You're welcome to do that. But on the specimen, it does contain the whole length of that grass stalk. Also highlights on a couple of these, if you've got extra space, it never hurts to put more individuals on the sheet. So grasses are notorious for this. They typically are, they're rather slender. So you could put several on there. We've got at least 1, 2, 3 inflorescences on here. And at the bottom, looks like we got about 5 underground portions of this same plant species. This one over here on the left, this is just one individual, but with a single bend there, we get from the roots all the way to the tip of this plant. So in these cases, it's not showing you exactly the perfect habit of the plant, but you can kind of reconstruct that based on the organization of the leaves moving up and then on the bent down portion as well. And then finally, getting into small herbaceous plants. There's some herbaceous plants that are so small that it'd be kind of a shame to put just one of them onto a herbarium sheet. So if you've got a rather small plant, often these are plants that grow in higher density. You might be able to grab a few of the same species and put them all together. So filling up the space, leaving some room for the label always, but otherwise pretty well filling up the sheet. And this has the upshot that a person could study variation within a population. So if you got these specimens, you could say, I know a range of variation of leaf size and flower number, whatever, with this species. I've got 7 individuals of that species right there. Okay, moving on to the topic of surveying. So we talked about how to make specimens, and part of understanding a habitat is making specimens and finding out just what's there. But it also can be important to have a strategy for determining what's there. So within a habitat, not every plant is growing in every part of that habitat. There might be species that grow isolated in one corner or another of the habitat. There might be a species that changes its abundance over time, over the space, say as you go up in elevation or maybe into a different soil type or in or out of the shade, can change the abundance of a species. And doing a thorough survey of a habitat can tell you more about the species and where they are found, more than just determining that they're present or absent. This last point on here, associations, is an important consideration as well. If you're doing an ecology type of study, you might be interested to know what plants are growing with what other plants. So relative abundances of 2 plant species next to each other could be useful to know. So we're going to have a chance to do some habitat surveying as part of this course. So we'll be doing a basic inventory to find out what's there, and then also using a strategy to determine more about where, sort of in the overall habitat, where species can be found. And one of the resources we'll use for that are topographic maps. So hopefully you've got some experience with topographic maps. I'll give you a little overview here, and I think I've got some more literature about these in other places. One of the important upshots of a topographic map is that it shows you elevation. So here we've got these lines, some thick and some thin. So this is 900, I think we're at 900 feet there. So 900 feet above sea level. And the lines go 900, 1, 2, 3, 4, and after 4 lines, there's another thick line that says 950. So this means that these lines are telling us 10 feet difference at everywhere that line shows up. And when you see lines close together, that would be a steep elevation. So we're going from 900 feet to 1,000 feet in rather short distance here. Other places, the lines are very spread out. So they're more spread out through here. It takes a lot longer to go from 900 to 850 than it did to go from 900 to 1,000. And then going from 850 to, boy, you can't even see the next one really. So 850, this would be, I guess, 840 here. Really gentles out. As you might expect, we've got some symbols in here that tell us that this is more of a flat area as well. So we've got blue for the watercolor, things labeled Whitewater Creek and Bluff Creek. And these symbols here are wetland symbols. So blue and green versions of this little wetland symbol here. We've got obviously standing water in the creek or moving water, and then wet areas around them, along with that very gentle slope elevation. You can kind of picture almost the kind of habitat that's in here. Other symbols in here, we've got green for more forested areas, and also the human element of these roads that are labeled on here. So roads and trails are all on the topographic map. So lots of really useful information contained on a topographic map. And you could actually sort of plan your survey strategy ahead of time using one of these. So for example, if you wanted to do a walk down the slope of elevation or up, you could plan to go down this part of the habitat and see if you've got different species at the bottom of this hill versus at the top of this hill. One of your possible strategies. So in general, I'd say one of probably the most general ways to survey a habitat is just to try and find out what all is there. And that's basically just called an inventory. And depending on how big your habitat is, you might not be able to walk the entirety of the habitat, but maybe you'll be able to find out that there are parts, sort of subsets of habitat within there that have different species. What I've shown here is a strategy for kind of estimating whether you've found enough species as you're going through the habitat. Have you counted everything? Is everything accounted for? Some species are going to be abundant, and you're going to count them pretty much anywhere you go in the habitat. I see a grass species that's pretty commonly found, very densely found throughout here. Other species like this white flowered plant, I don't see any of those in the background. This might be the only one for a little while. It might have taken some searching to find that one. So as you're going, you take an inventory and you record what's there, how much of any one plant there are. And you can build one of these, what's called a rarefaction curve. So what's shown here is kind of a visual way to, I'd say maybe illustrate what you probably would do anyway. How do you know how long you should keep counting new species? Well, if you take this gray line, and every time you count a new part of your habitat, or maybe for another 5 minutes of effort, you keep finding new species. Say you find 3 new species every time you count. These are new species. These are species above and beyond anything you've seen before. If you keep finding new stuff, you probably want to keep counting, because you don't know how far this is going to go until you reach the end of the count. By contrast, this darker line here finds a lot of new species initially, and then starts gradually tapering off. So we found 10 new species the first time, and then 20 species up on top of that the second time around, 15 new species, and 10, and so on. But eventually it gets to the point where it's like 3 new species, maybe no new species. And if this line flattens out or nearly flattens out, that means you've counted just about everything. So it's just a visual way to think about, if you keep looking, keep finding, you probably want to keep looking. If you're not finding anything new, you've probably got to the point where you've seen just about everything. So again, you're probably going to find the majority species pretty easily. But if you're trying to keep an eye out for the rare species, you want to keep looking for a while and make sure you've accounted for all possible places you could find them. So as far as surveying, the few strategies you could take, and we'll get a chance to explore some of these in the field. So just walking around kind of haphazardly and recording all the plant species you can find, that's one way to do it. That's a valid way to survey a habitat and find out what's there. A little more scientific, if you want to break it down by, say, parts of the habitat, subsets of the habitat, you can do more of a planned strategy. So one such plan is to sort of randomize yourself a grid system. This was a study we did where we randomized locations to look. So without being on the ground and deciding that one place was a nice place to look, we just planned ahead with a map of the habitat and said, we're going to look here, 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 we're going to look here. Then we found those places and we surveyed for species, and the map showing here shows us the places where this one species was found, Rudbeckia triloba, one of the brown-eyed Susans. And turns out that the relative abundance is the size of the circle here. And this plant was found relatively abundantly throughout this orange part of the habitat. So this was an area that was seeded, and it kind of made sense at the time, of course, that the place where the Rudbeckia seeds were sown is a place where we found adult Rudbeckia plants. Now this was still a few years after the seeds had been sown, so it's not necessarily guaranteed they would have grown there and stayed there or survived there. So it's shown that they are pretty abundant throughout orange, and one collection found in green here, maybe outside of where the seeds were originally planted. So at each of these points, we did what's called a quadrat. So quadrat is the spell check frequently wants to fix you on this one. Quadrat is different from quadrant, so it's intentional, and it's a square plot, and typical sizes are 1 meter by 1 meter, as shown here. That's a meter squared. or 10 meters by 10 meters. So depending on what you want to get for your output, it's kind of a benefit cost equation. If you want to find out, say you want to get a very fine scale look and account for small things, you might want to do one of these smaller plots. Each 1 by 1 meter plot is a lot quicker to do. Or if you want to get maybe more of a general inventory, you might do something that's 10 by 10. Takes a little longer for an individual 10 by 10, but there's a little less chance you might miss something in that larger quadrat. So the second bullet point here, randomizing or evenly spacing. So you can set up a grid of evenly spaced quadrats, or you could say use a computer, random number generator to find out random places to look. Randomizing is important because if you're in the field and you just choose to survey a place that looks interesting, you're probably going to be swayed by something like convenience, or you might be drawn in by an attractive flower. And it's not an objective way to survey habitat. So you want to remove your bias by planning where you're going to look before you see what's there. So if you have the time to plan one of these ahead of time, you can do what we did, where you determine where these spots are. So these are randomized points. They're not evenly spaced, but that's what we got for random number generating, and ended up surveying, we got 2, 4, 6, 8 locations in orange there. All right, so I mentioned maybe surveying in a habitat where you are going up or down a slope. Maybe you just want to get kind of a walk over distance kind of survey. That's the method known as transect. So a transect, usually these are linear, so just a straight line from one point to another. And along the way, you could either walk the line and just count everything that's, say, within 1 meter of the line. If you want to keep track of what plant species you find, you could do it that way. Another way to do that, as the second bullet point says, is to combine the quadrat method with the transect method. So you might take a straight line and every 10 meters survey a square meter. 10 meters, square meter, 10 meters, square meter. It's really, there's a lot of ways to tackle this kind of question. Depends on how much time you want to spend, depends on what your output interest, what you want for your output research to be. But overall, like I said, mostly what we'll be doing for this class is just getting kind of an inventory combined with abundance. And we'll get some practice at that as we go.